Welcome again. Today's video is about library preparation of DNA extracted from fur-bearer tissue samples provided by trappers and resource managers in Maine. A library contains DNA fragments from a single sample that are generally 300 to 500 base pairs long, with known DNA sequences added to each end to help them bind to the DNA sequencer. During extraction, the DNA is eluted as double-stranded large DNA molecules that are thousands to millions of base pairs long. For DNA, there are two base pair units, A pairs with T and G pairs with C. To cut the DNA to the correct length for our DNA sequencer to read, we use a Q-sonica. This machine produces a mechanical vibration which causes the formation and collapse of microscopic bubbles, releasing tremendous amounts of energy that shears the DNA. After shearing, we want most of the total DNA fragments in a sample to be below 500 base pairs in length. These graphs show the DNA fragment length on the x-axis and relative quantity of the DNA fragments on the y-axis. As you can see for the samples shown here, the shearing worked and all samples have greater than 85% of fragments below 500 base pairs. Now on to library preparation. The next step is N repair and A tailing. After shearing, DNA fragments might have one strand that is longer or shorter than the other. In this step, an enzyme repairs the overhangs or cuts them off, leaving a blunt end. Another enzyme then adds sticky A bases to the end of the molecule. Remember how A pairs with T? Well, this sticky A acts as a site for known DNA sequences called adapters, which have a complementary sticky T to bind to for the next step. Here I'm adding the DNA extract to a tube that has enzyme and buffer mix. Off camera, I'll briefly vortex and centrifuge the samples. Then the tubes are incubated at 20 degrees Celsius for 30 minutes and 65 degrees Celsius for another 30 minutes. This gives the enzymes enough time and the right temperatures to perform the repair and A-tailing. The next step is adapter ligation, where adapter sequences are added to both ends of the DNA fragments. These adapters provide binding sites during sequencing. Our fragments would not be sequenced without these adapter sequences, so this is a critical step in the library preparation process. Once the ligase enzyme and adapter mix is added, the reaction is incubated at 20 degrees Celsius for 30 minutes. After library preparation, we clean the ligated DNA to remove leftover enzymes and adapters that would inhibit sequencing. First, magnetic beads are added to the libraries, where they will bind to the DNA fragments. Once the beads are pelleted by the magnet, we'll discard the liquid, which should have all of the enzymes and unused adapters that bound to the other adapters instead of our DNA fragments. While the DNA is bound to the beads, they will be washed twice with 80% ethanol to rinse the DNA and remove any loosely bound adapters and enzymes. Here I'm adding ultra-pure water to the beads, which will disrupt the bond between the beads and the DNA fragments, releasing the DNA into the water where it can be used for the next step. Here are our finished libraries, and thank you for watching. The next step is indexing, where two unique DNA sequences are added to the libraries during a polymerase chain reaction.